London from the Weizmann Institute of Science, and he's going to be talking to us about using AI for screening covalent inhibitors. Nir, can you hear us?
I can. Can you see my slides?
Yes, we can. We might need the volume turned up slightly on your
Can you hear me
this
better
end.
now?
We could probably do that.
Okay.
Yeah, is that okay?
Yeah.
Thank you. Take it away.
Hi everyone. Sorry I can't be there in person. I'm anyway happy to talk and especially grateful for Frank for a great introduction to the topic. So our lab has been interested for a long time in the discovery of covalent inhibitors. Such compounds that are able to form a covalent bond with their target protein has a lot of advantages for chemical probes and potential drugs, such as increased biochemical efficiency. You typically need less of your compound to get the biological effect. They have longer duration of action. They stick around until the protein is degraded. And they can be very selective if they're targeting a residue such as a cysteine residue, which is only found in their target protein and not in close family members. For that reason, there are several successful drugs that were rationally designed to act as irreversible binders. It so happened that most of them are targeted against kinases, although KRAS is certainly a poster child of irreversible inhibitors. And so in my postdoc with Brian Choikat at UCSF, I developed a covalent virtual screening software based on classical methods. This was called docovalent, and over the years, we were able to prospectively apply it to discover some pretty potent covalent binders and validate the predicted poses by crystallography. As you can see here, a couple of examples. It was pretty accurate. But it was also very limited to very static structures. So docovalent does not include any flexibility in the receptor, even not at the side chain level, even not at the cysteine rotamer level. And all of these cases, we also had some sort of filter, such as for kinases, these compounds, we filtered them for compounds that bind the hinge region in the kinase, which is a characteristic of almost all kinase inhibitors. And so after we released this virtual screening pipeline, there was several other softwares that were being released, and benchmarks for their accuracy were started to be published. But as far as we could see, all of the benchmarks were for pose recapitulation and not for virtual screening performance, which is in our mind the more important sort of task. So it doesn't matter so much if you can accurately predict the pose of a ligand if you can't rank it first out of a series of a million or a billion compounds. And so we set out to build covalent virtual screening benchmark specifically tailored for covalent ligands. To do that, we borrowed the general idea from DUD or DUDE or DUDE-Z from the Choikat lab, where they computationally generate decoys that are matched in their physical chemical properties. So we start from an annotated covalent binder with an electrophile. We remove the electrophile. We generate property matched non-covalent analogs against using the same algorithms as the DUDE benchmark. So we have a few tens of decoys for each binder. We then reinstall the electrophile at a suitable position, and we filtered this set of compounds to end up with between 35 and 50 decoys that match in their chemical physical properties to the binders, and they're also drawn from commercially available molecules such that we know they're synthesizable, so the chemistry makes sense for these compounds. And when we looked for such binder sets for protein targets, almost all the targets we found were kinases. So our eventual data set, called CoValid, consists of 9 kinases and KRAS G12C, and altogether has about 40,000 binders and decoys. Here's the distribution of the physical chemical properties we matched. You can see for hydrogen bond donors, acceptor, charge, rotatable bonds, molecular weight, the decoys closely match that of the binders, so they shouldn't be easily discernible by a machine learning algorithm. Just one slide on the scoring function. So a lot of people mentioned AUCs, and AUCs are good to sort of discriminate between models over the entire distribution. But in virtual screening, we're especially interested in early enrichment, such as the enrichment factor that Frank mentioned. But we used a metric also kind of popularized by the Circuit Lab, where we take a log of the x-axis. And so in this, when we look at the area under curve in this semi-logarithmic plot, you have much more emphasis of the start of the area under the curve. And if you subtract the area of a random distribution, which is about 4.5%, you get a metric adjusted log AUC, which gives you 0% for random enrichment and 85.5% for perfect ranking of a hit list. And so first, I want to convince you that covalid has some applications, even for classical virtual screening. So we, for instance, there are adjustable parameters in covalent, such as the magnitude of the angles around the covalent bond that are formed that you can sample during docking. And we show that when we use the lowest value, we get a low average enrichment score over the set. When we increase the angle a little bit, we get a significant improvement, but we don't pay a lot in runtime. When we go to 10 degrees, again, improvement, but not such a big trade-off in time. But there's no sense in going to 20 degrees since we don't get any significant improvement and we're just wasting time. So this is an example of how we can optimize various covalent virtual screening software using this benchmark. And we did this for 3 classical physics-based covalent virtual screening software. One is DocCovalent, one is Doc6, and one is AutoDoc. And you can see here the results across the 10 different systems. And all of the softwares perform well in the sense that they give you values that are much better than random. So they do enrich. But then, as everyone knows, AlphaFold3 came into the picture. And while in the original publication, they did include a pose benchmark for 66 covalent ligands, which showed about 80% accuracy, we couldn't use this because the code was not available. The model weights were not available until about half a year later. But towards the end of 24, that was released. And so we can now use it on any arbitrary ligand. And here is an example of an AlphaFold3 model of a KRAS G12C covalent inhibitor that was not included in the training set. You can see that just from the sequence, as well as from the SMILES for the cofactor inhibitor and a magnesium ion, we get a very accurate pose recapitulation of the system. I should note that despite this very accurate model, both of the protein and of the ligand, actually when you focus around the covalent bond, it gives you non-physical bond length and angles. So it obviously didn't learn the physics of covalent bond formation, but it is able to compensate later in the next bond length and angle such that the overall pose of the ligand is very, very accurate. So something like Posebusters would have said this is a failed prediction, but in my mind, the fact that it was able to very accurately place the ligand, I think this is a very impressive prediction. So we wanted to see whether now we can use AlphaFold as a straight up covalent virtual screening method, and we try to run it on Covalid. As you may know, AlphaFold doesn't have an energy function per se, has different predicted confidence metrics. And so in the first iteration, we just modeled all of the complexes, protein ligand complexes in Covalid with AlphaFold, and then we minimized and rescored them using Rosetta. And you can already see in green that just by modeling with AlphaFold and rescoring with Rosetta, for half of the benchmark, we're way outperforming any physics-based covalent docking method. And we're about on par for the rest of the benchmark. This also means, in my mind, that the models produced by AlphaFold3 are realistic. So the fact that Rosetta doesn't give the binders bad scores and the Rosetta score so highly enriches for the binders tells me that the models that AlphaFold predicted make sense. They're not just scrambled coordinates that from a black box sense rank well. So after seeing that the model is realistic, we wanted to see whether any of the confidence metrics provided by AlphaFold3 are able to better rank than a physics-based energy function. But then we ran into the problem that the resolution of these confidence metrics is pretty low. For instance, if we look at PTM score for a few thousand decoys for BTK, you can see that 2,400 models get the exact same PTM score. So we can't say anything about the internal ranking of these 2,400 different models. Same goes for FGFR1. You get 900, 850 models with the same score. Still, just to get a broad picture, we use these metrics and sort of represent the results as ranges. So here you can see this range means the worst case scenario is if all of the models within one score were poorly ranked, and the decoys were ranked first. And the best case scenario is if for the same range the active binders were ranked first. So you can see the ranges are really big, as I already told you. But if you disregard this, big window, you can see performance is pretty good for most of these metrics, where it's specifically good for ligand-based metrics, such as ligand chain PTM in orange, which has a relatively smaller range, is consistently around 60% adjusted log AUC. If we just look at sort of the comparison to other software using ligand chain PTM, using the worst case scenario, it still works better in 9 out of the 10 cases. But by far the best metric we identified is this in brown MPAE, which is the minimal predicted alignment error. And you can see one advantage is the range for this metric is very small. And then also the performance is very high. So by minimal PAE, we outperform the physics-based method on all cases, and it nears perfect enrichment with an average adjusted log AUC of about 72%. Of course, the downside of using AlphaFold3 is that it's about 40-fold slower. So it takes about 4 minutes to run a single model, whereas the physics-based methods are very fast with about 6 to 8 seconds per model. But at least according to this benchmark, the upside is that you get very, very accurate ranking of the correct binders. Another nice property of MPAE is that it's independent on the system of the protein system. So for instance, when you run different ligand binding with Rosetta to different proteins, you can't really compare the scores so well because they're very system specific. But here you can see for each bin, we plot the percent of active binders that receive that MPAE score, and you can see how it's monotonically declining. So if you're below 0.9, you have above 90% probability of being an active binder. So this gives us sort of thresholds to now perhaps use in prospective virtual screening against new systems. Just to see that MPE doesn't succeed purely due to the computational nature of the decoys, we try to find reported ligands that are annotated decoys. That is, that they're similar to active binders, but they have very poor activity against the protein. We found 64 such ligands, and you can see for most of them, they get a very high MPAE, average MPAE of 1.5 angstrom, meaning that AlphaFold predicted them not to be active. Another interesting point is that NPAE cannot predict affinity. So in our hands, we don't see any correlation between the IC50 of the ligands and their MPAE score. So I think about it more as a binary threshold to tell you whether there's a high probability that this ligand would be a binder or not a binder, rather than a logistical regression to actually predict any type of affinity. When we first published this work as a preprint, of course, the major criticism we got is This is all memorization. I think this plot was already shown in this session, how co-folding methods improved as the targets are more similar to their training sets, and how this is not physics-based. In the last Q&A, someone mentioned, these negative examples, but you can mutate out the binding pocket and still get the prediction. And so we try to control for this not being a memorization artifact. The first thing we did is to purge the benchmark from any ligand that looks like any PDB ligand, but you can see even at very low Tanimoto coefficient threshold of 0.4, it doesn't really affect the performance against any system, even though the number of ligands that we can predict on decreases, the performance stays about the same. So it doesn't appear to be dependent on similarity to known crystallographic ligands. The other kind of computational proof or suggestion that it's not dependent is that there's no correlation between MPAE and similarity to any PDB ligands. You would expect if it's ranking based on known ligands, then the known ligands will have better scores, but this is not the case. But the ultimate test to prove that it's not memorization is to run a prospective screen. And the field is growing very fast, and I don't want to make bold claims, but I think this was the first sort of big prospective screen ran entirely with a co-folding method. We generated out of enamine real 900,000 ligands. We turned these into covalent ligands by installing an acrylamide on an available amine. We didn't the specification to choose the compounds were only molecular weight and diversity, so clustering. We didn't enrich the set towards, for instance, kinases or any specific protein target. So these are random molecules. We then made 900,000 models using AlphaFold3 against BTK as a model kinase with many known covalent binders. And we filtered these to only look at models with MPE below 0.9 angstrom. This resulted in 440 ligands. Then we filtered these to not have a Tanimoto coefficient bigger than 0.35, so they're completely dissimilar to any annotated BTK binder. This removed 50 ligands, so we're remaining with about 400 ligands that does not look like any BTK binder. And out of these, based on manual inspection and clustering for diversity, we selected 13 compounds for synthesis and evaluation. Of these 13 compounds, you can see here incubation at a large axis with BTK. 3 compounds showed close to 100% covalent labeling. These are the 3 compounds here. You can see their molecular structures. A few more compounds showed covalent labeling, but were not as striking. So we proceeded with further characterization of the 3 hits. First, I want to convince you that these hits are actually novel, chemically novel. So you can see for each compound on the left column, the 5 most similar compounds across all of Chembl, not just against BTK, but across all of Chembls. And I hope you agree that they look completely dissimilar to the hits. So these are genuinely chemically novel and could not be a memorization artifact, since there's nothing similar to be memorized. In a covalent labeling assay, when we incubate the compounds for 2 hours at increasing concentrations, you can see ibrutinib, our positive control. This is a covalent FDA-approved drug against BTK, labels very fast to 100%. Also, YS1, the top hit, labels very fast to 100%, and YS2 and YS3 are slightly poorer labelers. We see this even better when we do time response. So with 5 micromolar compound, 1 micromolar protein, you can see YS1 very rapidly reach 100% labeling, whereas it takes longer for YS2 and Y3. One possibility to cheat when doing covalent binding is to just choose very reactive compounds that would look in vitro like they're very potent, but then you'll suffer from selectivity problems in cells because the reactive compounds will react with everything. So to eliminate this, we ran a glutathione reactivity assay where we incubate the molecules with large excess of glutathione, a model thiol, and then we measure how much of the molecule was consumed. You can see here ibrutinib, after 48 hours with glutathione, is 80% consumed. But YS1, our top hit, is less than 20% consumed after 48 hours. So it's significantly less reactive than ibrutinib. Finally, we show that the compounds are able to thermally stabilize BTK after labeling. So this is the TM of BTK at around 48 degrees. Ibrutinib significantly stabilizes it by about 13 degrees, and YS1 is close at its heel with about a 9-degree stabilization. In an in vitro kinase activity assay, we see 30 nanomolar inhibition of YS1. Ibrutinib is much, much more potent. It's sub-nanomolar in this assay, but still 30 nanomolar with no medicinal chemistry optimization straight out of virtual screen is very impressive in my mind, specifically where the virtual screen in modern terms is considered small. People are now screening libraries of billions of compounds, and this was only around 1 million compounds. Another interesting fact is when we did the in vitro kinase activity assay against a mutant lacking the target cysteine, we completely lose our activity, showing that these compounds work through forming the predicted covalent bond with cysteine 481. These were also active in cells. In this assay, we activate signaling with incubation with an anti-IgM antibody. You can see BTK autophosphorylates, but then in a dose response, we can inhibit this autophosphorylation with an IC50 of about 100 nanomolar in cells. In terms of selectivity, we performed proteomic competition assay. This is an alkyne derivative of ibrutinib. So we pre-incubate with YS1, and then we incubate with this compound, and everything on the upper right quadrant are proteins that were competed out by YS1. So YS1 blocked ibrutinib binding, and you can see BTK and a close relative TEC were the only two kinases that were competed by YS1, with only a very small handful of other proteins out of more than 3,500 recognized, identified. This is a markedly selective proteomic profile. We ran a second proteomic profiling experiment with this promiscuous kinase binder. So you can see this probe enriches many more kinases, but still YS1 only competes for BTK, TEC, and to a very, very lower extent against this other kinase MARC4. In an in vitro kinase selectivity panel, we again see hits BTK and TEC are hits. BMX is virtually identical to BTK. Even ibrutinib cannot differentiate between the two. And JAK3 is an off-target that has a cysteine at the exact same location. So these are so-called the immediate suspects. There are only 3 other kinases that are inhibited, also not to a very high degree, so showing that these are very selective kinase inhibitors. In terms of the structure, this is the model that AlphaFold3 predicted for YS1 in the pocket of BTK. You can see the covalent bond here, the hydrogen bonds to the hinge region here, but it's kind of a unique model because most BTK inhibitors occupy this back pocket here, so extends behind the hinge region and go down towards this pocket, whereas YS1 kind of splits, makes the canonical hydrogen bonds to the hinge region, but sends half of the molecule beneath this flexible P loop. In collaboration with Amy Andrade's lab from Iowa State University, they were able to co-crystallize BTK with YS1, and you can see it's accurate to sub 1 angstrom, the model to the crystal structure, and indeed this chlorophenyl wedges below this P loop. I sort of stress this unique binding mode because there are 25 crystal structures of BTK with covalent binders, and none of them really makes this interaction. So it's possible that other kinase inhibitor in the PDB set that AlphaFold3 was trained on do make this interaction, but the ability of AlphaFold to kind of synthesize this and predict it for a ligand for this protein, I think demonstrates how practically useful it is, even if it just memorizes parts of ligands from other protein structures. We could also get a structure with YS2, which is markedly less potent, but it makes this non-canonical hinge interaction with BTK using a motif that you rarely see. And AlphaFold also got this prediction correctly. So to summarize, it seems like AlphaFold3 is a promising direction for covalent virtual screening despite its slow speed. We identified MPAE as a metric that dramatically outperformed any other method or energy ranking. We showed that prospective screening can identify novel chemistry, and I hope we refuted concerns of memorization, or at least showed it's practical for drug discovery purposes. We're currently looking into similar things to what Frank mentioned, making it more practical, for instance, by two-tier screening, running a billion virtual screen with classical methods, physics-based method, and then re-ranking the top couple of millions with AlphaFold3. We're improving our screening libraries. For instance, now we have a library that's specifically tailored for kinases with an enrichment of hinge binding motifs. So we think the hit rates in such library will be better. And we're looking into application for non-covalent screening. So two final points I want to make in this sense. The first is, started the talk by introducing doc-covalent and showed that we were able to find covalent kinase inhibitors with it. And now we're also able to find covalent kinase inhibitors. So what changed? So flexibility is a major thing that changed. So for instance, YS1, you can see here in white, clashes with this phenylalanine that's seen in the crystal structure of BTK with ibrutinib. And doc-covalent ranked YS1 at place 150,000 instead of the top 400. So the ability of AlphaFold to model flexibility during the docking, I think, is crucial for getting novel chemistry and much more accurate results. And then we're trying to apply this now to much more challenging cysteines, for instance, cysteines on flexible loops that AlphaFold can co-model the loop flexibility during the modeling of the kinase inhibitor. And we think this is an application where AlphaFold would really shine compared to a physics-based model. And then in terms of non-covalent docking, we ran AlphaFold on DUDZ. So these are 40 different targets that have many more ligands and much more diverse than just kinases. You can see on this more diverse data set, it doesn't show the near perfect classification that we got for covalid, but still it's much better than either version of doc that was reported for this data set. And I know there are really in the recent months or so, there were preprints from two different virtual screening groups that say that they also see better results on DUDZ, but they don't think this sort of translates to virtual screening, and some other prospective virtual screening are starting to come also for non-covalent methods, and I'm curious to see how this sort of progresses in the future. With that, I'd like to thank you for your attention. I'd like to thank Yoav Shamir, which single-handedly did all the computational work you saw in this presentation. Ronen and Adi helped in the experimental side. Amy Andriotti's group with the crystallography, and the funding agency is happy to take any questions.
Thank you very much. So do we have a quick question? Please say who you are.
Thanks for the lovely presentation. My name is Sofia Caria from Evotech. You were talking on the end of your presentation about the flexibility that is now accounted on AlphaFold3. My question is, how much bias is that by the availability on the PDB? Meaning, if you have a structure of a protein which is not very well represented, and especially if it's highly flexible, how good is the prediction currently for AlphaFold3, either covalent or non-covalent compounds?
Yeah. So
thank you.
So thanks for the question. I think it's a good point, a good place to add a disclaimer. I think the great performance I showed for AlphaFold3 will only hold for very precedented pockets, such as kinases, such as nuclear hormone receptors, places where it's seen a lot of ligand protein complexes. We do have evidence on proprietary structure that we have in the lab that are not in the PDB and are not precedented in the PDB. It wasn't able to model cryptic pockets or shallow non-orthosteric pockets. So I think you should restrict your screening to well-precedented orthosteric pockets, but this still leaves you with a huge space. You know, kinases, maybe only a few tens of kinases have known ligands where there are more than 500 different kinases. So in terms of flexibility, I think as long as there are a lot of data on binding to the orthosteric pocket, it learned enough about protein dynamics and sort of loop conformation from the entire PDB to be able to accurately model the loop conformation, but it needs all of these data, broad data on the orthosteric pocket to be accurate on your system.
Okay, thank you very much for that. So I'm conscious that lunch is outside, so I think in the interest of time, we're going to have to stop the questions there. Please, can we please thank Nir and all of our speakers again? If you have questions for any of the speakers, please ask them on Slack. You might want to at the speaker to give them the best chance of seeing it so they don't have to scroll through all the messages. Thank you very much.
Bye-bye.